Okay, so welcome to this next video in which we are discussing the HIV life cycle and antiretroviral drugs. Okay, so we have got now to the point where the viral envelope has fused with the plasma membrane of the cell. Okay. And then what's happened is the P17 matrix has broken down and the nucleocapsid is now passing into the cytoplasm of the cell. Okay? What will then follow is that the capsid, the P24 capsid, will break down, and that will release then into the cytoplasm of the cell the two pieces of single-stranded positive sense RNA here. Okay, so let's colour those in green. Okay, and then also the three enzymes that come too, so let's have these in. Okay, so we've drawn these previously as just little circles, so we'll stick to that for now. Okay, so here they are. So remember, the three enzymes are reverse transcriptase, which is the first one that's going to act now. Okay, then we also have, and let me just move this to the side a little bit. Okay, we also then have integrase. Okay, and then finally HIV protease. Okay, so what's now going to happen is that reverse transcriptase is going to act on the single-stranded positive sense RNA here, and it's going to produce a piece of DNA from this RNA. Okay, so for our purposes now, we don't even need to think about the fact that there are two of these. We only need one for what's going to happen now, but two of them will just mean that the thing happens twice, effectively. Right, so what will now happen is if we take this single-stranded piece of RNA here, which we're colouring in green, what we're going to do is reverse transcriptase basically will synthesize a piece of DNA that is complementary to this. Okay, so it will synthesize a single-stranded piece of DNA that is complementary to the positive sense single-stranded RNA here. Okay, now which piece of DNA will this effectively be? Because remember, we were talking about the fact that there's always a coding strand and a non-coding strand. Well, this one is complementary to the positive sense RNA. The positive sense RNA is what can be translated, so this effectively is the coding strand, basically. Okay, so you will produce this complementary piece of DNA that is effectively the coding strand of the DNA that would make this RNA. Okay, and this is called cDNA. This single-stranded piece of DNA is called cDNA, and the C there stands for the complementary piece of DNA. Okay, and it is reverse transcriptase which is capable of synthesizing this piece of cDNA from the piece of RNA. So reverse transcriptase is an example of an enzyme which falls into the category of RNA-dependent DNA polymerases. And in a normal human cell, we don't have RNA-dependent DNA polymerases. So this means an enzyme which is capable of producing a DNA polymer from RNA. We don't usually have enzymes that can do that. We don't need enzymes that can do that. This is something that, therefore, the HIV variant needs to bring with it. So reverse transcriptase is extremely important. Okay, and we'll see drugs in a moment when I talk about them that block reverse transcriptase and stop the synthesis of this complementary DNA strand from the single-stranded RNA. Okay, right. Now what will happen excuse me, is you will break down the mRNA that is attached to this DNA, and then what will happen is enzymes within the cell will perform DNA-dependent DNA polymerase. Okay, so you'll use an enzyme that comes in the category of, excuse me again, DNA-dependent DNA polymerases, which is just basically any enzyme which synthesizes a complementary DNA strand from another DNA strand. So we've got now one DNA strand, which is this coding strand, and remember we've broken down the piece of RNA so that it's no longer bound to this piece of cDNA. And then what we'll do is we'll synthesize the complementary DNA strand to this, like so. Okay, and I'll colour this in pink here. So in pink, this is then the non-coding DNA strand. Okay, so this is the non-coding strand. Right. Okay, so we now have a piece of double-stranded DNA which contains the same genetic information as the original single-stranded piece of RNA that was inside the nuclear capsid of the HIV variant. And this piece of double-stranded DNA is then called the Pro Virus. Okay, what will now happen is this Pro Virus will be inserted into the DNA of the human cell. So basically, what will happen is you will go up to some chromosome within the human cell, you will cut the chromosome, you will make a double strand break. Okay, so let's say this is our chromosome. What we will do is we'll make a double strand break, and then we'll insert this into here, so that what you now have is this HIV Pro Virus inserted into the human chromosome. So let me colour it in so that we can easily see it here. So this is this coding strand, and this is this non-coding strand in pink here. Okay, so now the HIV Pro Virus is inserted into the human genome, and the enzyme which does this is the enzyme integrase. Okay, so this is an essential part of the HIV life cycle, that the single-stranded RNA that we had within the nuclear capsid is converted into a provirus, and then the provirus is inserted into the human genome. Okay, and for this to occur, we need the HIV integrase and also the HIV reverse transcriptase to be functioning. Therefore, these enzymes are key drug targets, basically. So, what I now want to discuss is reverse transcriptase inhibitors and also integrase inhibitors. So, let's start with reverse transcriptase inhibitors. Okay, so there are two classes of reverse transcriptase inhibitors. There are nucleoside analog reverse transcriptase inhibitors, and then there are non-nucleoside analog reverse transcriptase inhibitors. Okay, so you can divide reverse transcriptase inhibitors into two main families. The nucleoside analog reverse transcriptase inhibitors, and remember, a nucleoside is just the name for a organic base bound to a ribose sugar. Okay, so let me just draw a little picture of that. So basically, a nucleoside means a ribose sugar, like so. So this is the ribose sugar bound to some organic base here. So this is the base. So that could be adenine, guanine, cytosine, thymine, uracil potentially, and then we've got a ribose sugar here. And it's very important that you understand that that's a ribose sugar, not a deoxyribose sugar. If we're talking about deoxyribose there, you'd call that a deoxynucleoside. Okay, so here we have in blue the ribose, and the organic base we'll have in green. So nucleoside analog reverse transcriptase inhibitors are structures that are similar to nucleosides, and of course this enzyme is going to be working with structures that are similar to this because it's producing a piece of DNA. Okay, so these sort of drugs, it's understandable how they might go into the active site and bind there and then block the activity of the enzyme. Okay, but there's also a family of reverse transcriptase inhibitors, which is the non-nucleoside analog reverse transcriptase inhibitors. Okay, so let's discuss a few very famous examples of each of these groups. So there is one drug that you cannot talk about, the antiretroviral drugs, and not include this drug. This was the first drug that was produced against HIV. And during the HIV crisis in the 1980s and into the 1990s, this drug was prescribed by the bucket load, basically. You know, when HIV was a death sentence, and there was a time when it was a death sentence, basically you'd come into your doctor's office with this disease and they would just give you handbags full of this drug, and that was all they could do for you, basically. And it would prolong your life, but it wouldn't stop the disease eventually killing you. Now with combination therapies where we use loads of drugs all together, we can actually stop HIV killing you, and we can return your life expectancy to normal. Okay, so this drug, however, is still used in the combination therapy, so it still has an important part in antiretroviral therapy. Okay, so this drug is zidovudine. Okay, but its old name, its more famous name maybe, is AZT. Okay, and AZT stands for azidofimidine. Okay, so the AZ is for azido, and then the T is for thymidine. Okay, so it's got a very similar structure to the thymidine, okay, which is a deoxynucleoside. Okay, so thymidine is the name for thymine bound to deoxyribose. This is an odd naming system because thymidine, you would think it was ribose with thymine. However, thymidine is actually deoxyribose with thymine because, of course, thymine isn't included in RNA, it's only included in DNA, so it seems logical not to use this nice word that we have here, thymidine, to describe thymine bound to ribose, which isn't actually used for anything, but is only a precursor to making thymine bound to deoxyribose, which is then used in DNA. So thymidine, this means deoxyribose plus thymine, the organic-based thymine. Okay, so it's actually a deoxynucleoside, but obviously it's similar to a nucleoside. Right, so that's a very, very famous drug. Now let's turn our attention to some of these non-nucleoside analogues. So two non-nucleoside analogues are nevirapine and also efavirenz. Okay, so both of these are very famous drugs as well and are commonly used in cocktails of drugs that will be given to people with HIV. Okay, so all of these drugs work by blocking the reverse transcriptase enzyme and stopping it from being able to function. And when you stop that enzyme from being able to function, then you stop the virus from being able to turn its single-stranded RNA genome into a provirus. And without the provirus, the virus cannot replicate within the human cell. And therefore, you've halted its life cycle. Okay, next up, we're going to talk about integrase inhibitors. Okay, or rather I'm going to talk about one integrase inhibitor. I'm only going to give you the name of one, the main one, but the principle is what's important. Okay, so these are drugs which will bind to the enzyme integrase and stop it from being able to work. Okay, so the main example of an integrase inhibitor is the drug raltegravir. Okay, so raltegravir. So other drugs in this category, they all end with this gravir basically. So if you see other ones, you'll be able to recognize them by the ending gravir. Okay, so this drug will bind to integrase, stop it from functioning, and hence the provirus, the viral provirus, will not be able to be implanted into the human genome, and that blocks the life cycle as well because the provirus needs to be put into the human genome. Okay, so we've now got to the stage then where we have this viral provirus within the human genome. We are now ready to look at how the HIV virus is actually going to start producing new HIV virions. But before we do that, I just want to discuss the reason that HIV viruses cannot infect naive CD4 positive T cells. Okay, so basically one thing that we found out basically is that we know that HIV can in principle infect CD4 positive T cells, but in reality it's only a successful infection in some CD4 positive T cells, and these are the ones which have been activated. Okay, so ones where professional antigen-presenting cells have come and presented antigen to them and activated them. So activated T cells and also the memory T cells, because those have been activated but have gone into a memory state. Okay, but you cannot, basically, HIV cannot infect naive T cells. It can get into them, but it can't successfully complete its life cycle within them. And I now want to discuss the reason why. Basically, the reason is that naive T cells, okay, so T cells that are potentially sitting in the lymphocytes, the lymph nodes rather, and have never seen their antigen, these have a enzyme which is given the rather cumbersome name ApoBEC3G, ApoBEC3G. Okay, now what does this stand for? Well, it's even more cumbersome what it stands for. The ApoB stands for apolipoprotein B, okay, and then the rest of it stands for mRNA editing enzyme catalytic polypeptide-like editing complex 3, which is a complete mouthful, but I'm going to write it down nonetheless. So this is apolipoprotein B, mRNA editing enzyme catalytic polypeptide-like editing complex 3. Editing enzyme catalytic polypeptide. They've just sort of thrown words together, really. Catalytic polypeptide, and then like editing complex 3. Okay, so like, and now am I not going to be able to fit it in there? Okay, so I'll go up here, like editing complex 3. Okay, right, so ApoBEC3G is this protein, this enzyme, which is operational in naive T cells, but not in the activated T cells or the memory T cells. Okay, and let me explain what it does, basically. It is a type of enzyme known as a cytidine deaminase. Okay, and basically cytidine deaminase enzymes convert, they basically convert cytosine to uracil in DNA. Okay, so where you have cytosine organic bases in DNA, cytidine deaminase enzymes can convert those to uracil. Okay, and basically what's going to happen is this specific enzyme, this specific cytidine deaminase, ApoBEC3G, is going to specifically start converting cytosine residues to uracil residues within the HIV Pro Virus. So let's show our HIV Pro Virus implanted into the human genome here. Okay, so here it is, like so. And remember, we have a coding strand and a non-coding strand. So the non-coding strand we were showing in vivid purple, and the coding strand we were showing in turquoise here. Basically, where you have cytosine residues in this DNA, what you're going to do is you're going to convert those cytosine residues to uracil residues. Now, you don't usually have uracil organic bases in DNA, okay? And what this leads to is it leads to problems with the expression of these genes, basically. They can't be they can't be copied by they can't be transcribed properly, basically, by the RNA polymerase enzyme. Okay, so it stops the enzyme it stops the provirus from actually being able to replicate within the cell. Okay, so this ApoBEC3G in the naive T cells is fantastic at stopping the virus from being able to actually replicate within those naive T cells. Now, basically, when T cells activate, okay, what happens is this ApoBEC3G is incorporated into a larger protein complex. So here it is as a monomer, as a functional enzyme, but basically upon T cell activation what happens is loads of other proteins join onto it, okay, and it potentially gets some other function now, okay, but the point is that it loses its function at being able to convert cytosine residues to uracil residues in the HIV Pro Virus. Okay, and therefore you lose your resistance. So that is why naive T cells cannot be infected with HIV, although activated T cells and memory T cells can be infected by HIV. And I want to stress that HIV can get into both of them, it just can't replicate in naive T cells, whereas it can replicate in the activated T cells and the memory T cells. Okay, and we'll continue this discussion in the next video.